It's the Bird Emergency, the show where we talk about bird scientists, rare birds, threatened birds, things that might threaten birds, and the people who are trying to stop all those various threats. And I'm Grant Williams. I might add, I'm a bird nerd. I'm not a scientist. So please bear that in mind. Michelle Wille is joining me today. And Michelle is currently working out of the University of Sydney, but Michelle's an expert in bird flu. So we're going to talk about the potential threats of bird flu, but also where Michelle has been studying bird flu and what she's been looking at. Welcome, Michelle. Thank you very much for being part of the show.
Thanks for the invitation. Thanks for having me.
Michelle, I couldn't resist what I saw in some of the material that you sent me when I reached out to you. Penguins. Bird flu in penguins. Let's start with the penguins.
Sure, let's start with the penguins. Penguins are awesome.
So, what species, where, and the bit I'm fascinated about, how do you study influenza in birds in the Antarctic or the subantarctic?
So, I guess we study influenza in birds in the subantarctic and the Arctic the same way we do everywhere else. And that involves going to these really cool places, catching birds and taking samples. And so the kind of samples we take, they're called cloacal samples. So they're a sample of the butt of the bird. So birds are like reptiles. They're pretty cool. They just have one hole at the end. So the reproductive tract comes out there and the gastrointestinal tract comes out there and the urogenital tract. So it all comes out in one place. And so we take a sample of all the sort of cells there to capture everything. And then after we finish taking our samples, everything needs to get back to the lab eventually, and we'll screen for those samples. Now, the interesting thing about Antarctica that you might not guess, it's actually pretty hard to keep samples cold in Antarctica. So these samples that we take are temperature sensitive. They shouldn't fluctuate in temperature too much. We like to get them into these super ultra cold freezers as soon as possible. But, different locations in Antarctica don't all have ultra cold freezers. It's not -80 out there all the time. And so a striking problem of working down there is the logistics behind keeping these samples really cold so that we can use them again.
So is that because you take the samples, you're working down there in summer, and what's a nice summer's day in Antarctica? What's the temperature you'd be working in?
It was a few years now since I was down there. It's 2012. But I think I remember most days being like 5 or 6 degrees where we were working. So it was warmer. There were some periods where it was colder, where we had storms come through. But I was working on the Antarctic Peninsula, so not the deep freeze that is below the Arctic Circle. So it was actually quite pleasant to work down there most days.
So you're taking your samples. So you're wiping the bird's bum, basically, and you're working with the long test tube and the long swab. Is that how it works?
We just have a little tube. It's usually regular sort of.
Yep. And you've got like a little foam esky or something, a little that you put the samples in, and you've had to pack that with dry ice, I would guess, if you've got to get them down to super low temperatures.
Yeah, so usually in the field we would just pack it with an ice pack, and then when we get back to our base wherever we're working, we're either putting them at 4 degrees, or if we have a super cold freezer, we're putting them in a -80 freezer. And then of course to transport them from Antarctica to wherever our lab would be, we transport them on dry ice. Yeah.
So it just explain how it's difficult to keep them at the right temperature if it's only 4 degrees, and is that cold enough?
So for short term, it's all right just for a few days, but we need to get the samples into an ultra cool freezer or cold temperature within a few days. But the problem is less that 4 degrees. It's more keeping the samples at a constant temperature and not fluctuating. So for example, we'd go to a place for 2 weeks and then pack up and then have to transport everything to go somewhere else again. And so it was surprisingly hard to keep samples in good condition.
And at what kind of temperature do they start to degrade?
Well, within a week at 4 degrees, they would definitely start to degrade. Surprisingly, viruses, especially RNA-viruses, are pretty wimpy. If you warm them up a few too many times or thaw them a few too many times, they degrade pretty fast. Yeah, so that's why we use ultra-cool freezers normally.
So as I understand it, there's 2 major types of bird flu. So Can you walk us through those? And then do penguins have pandemics, influenza pandemics, mass kill events?
I'll answer your question in two parts. The first part is about influenza in birds. And so normally that's divided into kind of these two broad categories. The first category is the kind that you've probably read about in the news. This is where a lot of chickens die. This is where we're afraid of these viruses getting into people. And these are usually called highly pathogenic avian influenza. So pathogenic and highly pathogenic means it causes disease or death. And this is commonly seen in poultry, but it's usually, and I say usually, quite rare in wild birds, except for the last few years where there have been wild birds implicated in the spread of highly pathogenic. But for the most part, what wild birds have is called low pathogenic avian influenza. So these are viruses that don't cause any signs of disease and they don't cause mass mortality. And we think that these viruses have been evolving with birds for millennia, such that they've co-evolved, so they don't really cause disease. So that means that in the Northern Hemisphere, in the autumn, you can go sample ducks, and 20 to 30% of ducks will have low pathogenic avian influenza, but they won't have any signs of disease and they won't be dying. So what we found in penguins is this low pathogenic avian influenza virus. That means that we didn't see any sick penguins. We didn't sample sick penguins. We sample healthy penguins, and these penguins happen to have quite a lot of viruses, including these low pathogenic avian. So as for pandemics and disease in penguins, there have been a number of incidents of sort of mortality events or disease events in penguins in Antarctica and sub-Antarctica. To date, none of them are tied to influenza. They're tied to other things. So, for example, there was an outbreak of something called avian cholera. It's a bacterial disease some years ago, maybe 10 or 15 years ago. I don't remember the details. And there are incidences of feather loss or a number of other diseases that happen, but they're not due to influenza to date.
Do you think this strain, the pathogenic strain, has evolved with wild birds, or is it as birds have become domesticated and husbandry techniques have kept them at close quarters and all sharing food receptacles and things like that? Or has the pathogenic strain always been out there?
So it's probably really tightly linked to poultry production and intensification of farming. And these pathogenic strains have evolved more than once. They evolve repeatedly. So, for example, last year in Victoria, we had a really big outbreak of avian influenza in poultry in Victoria. And so what we believe is that it was a low pathogenic virus that was in a wild bird that entered poultry production. And if you have tens of thousands of birds that are very close together, many of them are young birds, essentially what you're doing is inserting a virus into the system, and it replicates and replicates and replicates and replicates, and it acquires mutations. And some of these key mutations allow for the virus to go from this low pathogenic phase to the highly pathogenic phase. And then we'd start to see morbidity or disease and death associated with that. So there have been a number of events around the world in the last 15 years where we've seen the emergence or sort of these highly pathogenic viruses arising in poultry production. And so it has to do with intensive agriculture, but it also has to do with chickens, we think, due to some of the funny features of their immune systems, although I'm not an immunologist. And so it's not that these highly pathogenic viruses have been evolving with wild birds. It's something that's tightly linked to poultry production.
And the second part of my question was whether there's mass kill events of influenza. Do any of the bird populations that you know of that you've looked at have pandemics?
So I think that we've seen mass mortality events in wild birds associated with a number of diseases, especially many years ago, actually still ongoing today, is sometimes these highly pathogenic viruses escape poultry production and re-enter the wild bird reservoir. And we've often seen a lot of mortality associated with that. And so this is many years ago now, maybe 15 years ago. For example, there is this big sort of lake in national park, an important stopover site for wild birds in China, where they found a lot of dead wild birds, including bar-headed geese. And that was due to these this highly pathogenic avian influenza that had escaped from poultry and into wild birds. I know that the Netherlands in the last few years has seen quite a lot of mortality in widgeons. It's a duck species called a widgeon, and due to these highly pathogenic strains that are now found in wild birds.
Okay, so the threat is, as so often, it's our practices and using birds as food sources or as pets and keeping them in close quarters is the threat to the wild populations, that the pathogenic virus is likely to move from birds that are hanging around with us into wild populations and finding susceptible species.
That's always a concern. Not all poultry viruses have re-emerged in wild birds. Some of them have decided to stay in poultry. But yeah, lots of viruses that wild birds have, they would have had for a long time and largely co-evolved with. So in my research, not only do I work on avian influenza, I work on lots of other viruses. And we're finding that wild birds have quite a lot of viruses in them. But these are healthy birds. And so presumably they've had these viruses for a long time. But the concern is that sort of these poultry viruses may re-emerge in wild birds, causing disease and death.
So in most cases, it's like the bird having a cold, but
we're not even convinced that in most cases. Like the birds that we sample are birds that are doing their regular thing. They're
just symptomatic.
There's no observable signs of disease. So we tend to not say asymptomatic because maybe we can't record or see everything that the bird is. There's nothing obvious that the bird is doing that would make us think that it's sick or not feeling well. These are birds that would be captured in a flock or in traps with other birds. They're just doing their regular thing. And so in my research, I don't think we've sampled any birds and not found a virus in them. All birds have viruses.
Are there any others that you think are potentially a concern for any wild populations that, apart from avian influenza A and B, we've talked about? We've talked about beak and feather disease.
Yeah, that's the one that comes to mind for sure, because I think big parrot populations can probably support having some individuals die of that disease, but there's always the concern, of course, when it gets into rare species.
Now, dear listener, this is where I prove that I'm not a scientist. Okay, this is when I get to ask a really dumb question.
Sure, it's not dumb.
So I live in the urban area, and there's lots of pigeons near me, and I often see lots of silver gulls in the city in Melbourne. And I see a lot of them with what appear to be deformed feet. Now, I used to believe when I was a child that the seagulls had deformed feet because sharks came and attacked their feet when they were swimming on the water. But pigeons don't do that. And I see a lot of really severely deformed feet in pigeons. Can you hazard a guess at what are causing these deformities?
So let's start by saying that I'm going to hazard a guess from my memory from somewhere. I might give you the wrong answer because I've misremembered. But I think that the disease is called clench claw, and it's seen in passerines, not just pigeons, but passerines and things. And I actually have a colleague who had a paper out last year about maybe one of the causes of clench claw. And I don't remember what she found, but I think she did find a virus associated with whichever bird species she looked at. Again, I don't remember the details, but there's probably something causing that. It's not quite clench claw, but I also read a really interesting paper this morning about a virus called it's a virus in the family Picornaviridae, that's a picornavirus, causing these beak deformities in North American birds called virus. And they found it in many North American bird species now. So these kinds of weird things may very well be due to viruses, which we may or may not have described.
Yeah. Not often I see pigeons with deformed beaks and sometimes gulls, but I've seen lots of deformed beaks in finches, in cage birds, canaries, and things like that. So that's viruses doing their things too?
Yeah, I think so. So there are a number of viruses that cause disease, and there are lots of these viruses that we find that are not associated with obvious disease. What they're doing, we don't really know.
Now, you did a lot of work studying mallards, ducks. So let's talk about that work. I'm really interested in how you do the work as much as what you learned. So Can you step us through how you design a study and what was the question that you were attempting to answer or the problem that you were attempting to solve? And just let it fly.
So I did my PhD in Sweden, and I worked in a lab that was using Mallards as a model for understanding the ecology and evolution of influenza viruses. So it was a really cool place to work because my lab was based in a small town called Kalmar, which is quite close to a bird observatory called Ottenby. It's one of the longest running bird observatories in Europe. It's been there for a very long time. And adjacent to the bird observatory is a very large duck trap that's been there for a very long time. I think when the duck trap was originally set up, I might have misremembered, I think it was used for hunting. This is like 40, 50 years ago. But since about 2002, it's been used for research. So there are people that are employed through both the observatory and through our research group who would go and use this duck trap to catch ducks every day.
Excuse me, Michelle. I have to ask, is the duck trap like a really big box resting on a stick with a string attached to it, and you wait until the duck goes under it and you pull the string?
It was actually one of the most sophisticated duck traps I've ever worked with. It's quite massive. This duck trap is like, let's try to think, maybe 10 meters by 30 meters or something. It's a really big duck trap. And so part of the trap is in the water and part of the trap is on land, and it's like a big mesh cage. And there's little doors in the sort of the seaward side of the duck trap like this. So the ducks can find their way in, but because of the shape, they can't figure out how to get out. And there's a compartment within this duck trap wherein we actually put ducks used to lure the other ducks in. We call them lure ducks, but the name for them obviously is sentinel ducks. So there's 10 female ducks that live in the trap, and it's a big trap, right? Very big. They've got space to swim and forage, and there's a bit of land for them to hang out. And think aviary, really big. Yeah, and we would put grain in the trap every day for when there was water. Of course, in the winter, it's all frozen over.
And within the aviary, for want of a better descriptive term, you have a series of smaller little cages with really sexy lady ducks, and they're going whack, 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 whack. And there's a banquet on offer, and it's like a clay pot. They can get in, the ducks can get in through the funnel, but of course they can't work out that there's this little narrow hole to get out. So there's no trapdoors. There's nothing like that.
No, and on a really good day, we could catch 100 ducks in that trap. So it was a very efficient duck trap that didn't really require a lot of work.
So the sexy lady ducks have done their job as the decoys or sentinels. And what do you do with the captured ducks? How long are they deprived of their liberty?
Well, so most of the ducks would come in around or just before sunrise, and then we would empty the trap usually not long after sunrise. So in summertime, the duck trappers and scientists would be there pretty early in the morning to go and get all the ducks out. And it usually didn't take very long. And so in that system, all the ducks were then put into these single-use cardboard boxes. So sometimes it was 100 cardboard boxes in a day, and sometimes it was far less. But the nice thing about cardboard boxes is the ducks tend to defecate in the boxes, so that we could just take a sample of the poo instead of having to take vehicle samples. So that worked really well. And sometimes when we were out there, we would take a blood sample as well. There's a little bit about what's happened in the past with the duck using antibodies. And then we would let the ducks go, except, of course, the sentinel ducks, which we would also sample, and they would go back.
Now, you're part of the Pint of Science team, so you would be aware of the show we did with Toby Ross. And Toby, in conjunction with where was the lab he was working with? Were they in Norway? Where the blood samples are sent? Is your work doing anything cooperatively with other teams when you're taking blood samples? Are you having to send your work off to Norway, your samples off to Norway?
So the really fun thing about my job is I get to wear 3 hats. I get to be an ornithologist and go catch birds, and then I get to be a virologist and I sit in the lab and I actually process all my own samples. Toby Ross is actually a colleague of mine, and so a number of the blood samples I've collected in Australia have gone towards his project, and some of the samples that he's collected have gone towards some of my projects. So that's quite fun, but throughout all the places I've worked, both the blood samples and the collected samples are samples that I've processed in the lab wherever But sometimes we've given extra samples to other people doing other projects that maybe process elsewhere if there's leftovers. Yeah. Once we collect all the samples from our ducks or whatever we're sampling, they obviously would go then to the lab for processing.
And what did you discover?
So during my PhD, I was quite interested in understanding how influenza viruses stay in populations over many years and within years. So these are questions, evolutionary questions about the virus. So we did a few studies. The first study was looking at within an autumn. So again, in the Northern Hemisphere, autumn is the time of the year when we find that ducks have really high prevalence of virus. So you could go sample a duck in September, October, like 20% of the ducks might have an influenza virus. So in the beginning, I told you we had these low pathogenic influenza viruses and highly pathogenic influenza viruses. Within this thing called low pathogenic influenza virus, there's actually 16 sort of more divisions. And these are 16 different virus strains. They're really different from each other. In humans, we tend to have H1 and H3. These are the human subtypes or strains. In birds, there's 16. So it's pretty incredible. And influenza viruses do this really annoying thing where they like to mix and match the bits of their genomes. So again, this is where human pandemics come from. Remember you heard about swine flu? It was actually the mixture of viruses from pigs and from people that came together that created a pandemic. So in birds, we have these 16 subtypes and they tend to mix and match with themselves all the time. So I did a study with colleagues trying to understand what dictated the mixing and matching and which viruses mixed with which other viruses. And so we found that in birds, there's a lot of mixing and matching, and they tend to mix and match with whatever else is co-circulating at the same time. So we would have an H1 and an H6, and then an H4 and H5 that were mixing and matching. And so that was really interesting. So it shows you how the virus can evolve in a really short time frame, such that even if the birds develop immunity in a short time frame, they can continue to appear different and new and circulate for much longer. We also did this study where we tried to look at what happened over 10 years. So for example, how does something like H4 appear in the population year after year? And we figured out that within this strain category, there was more subcategories, and we found a different substrain every year that came through the population. So that was pretty interesting as well.
And if most cases the birds don't appear to be sick, does that mean the birds are winning the war against the viruses? And actually, I'm tempted to ask you the worst question I think anyone can ask about viruses is, why aren't they alive?
Okay, so that's the second part. So there is this interesting discussion in disease ecology. It's called whether the host resists or tolerates infections. And so to resist is to mount a really big immune response. Sometimes when we feel unwell, it's because our body is mounting a really big immune response, trying to kill whatever we've got, and we feel unwell about it. On the other side of that spectrum, or the flip side of that, is they just tolerate the infection. And I think this is what wild birds are doing with these influenza viruses. They're just tolerating. So I'm not really sure whether they've won the war or they've just found a way to keep the peace. And then as for why viruses are not alive, it's a biological question about the fact that viruses can't make more viruses without a host. They just can't. They're so small. They just have the little bits of their genome that they need, and then they'll take over the cell of something else to make more of themselves. Without hosts, viruses can't make more viruses. It's so bad that the smallest virus only has 1,600 nucleotides, A's, C's, T's, and G's. It doesn't even make a protein to replicate that. It just makes a bit of the outside and then steals bits from other viruses and from the host. And so they're actually really efficient, given that they can't make more of themselves.
I've always struggled to understand. I know, but then you think, well, why can't a rock
But a virus can replicate, it just needs
The rocks and trying to replicate the rocks. But yeah, no. It's one of those classic questions, isn't it? What's
but not alive.
No, that's right. That's right. So sorry everyone for that stupid question, but
No, why don't we classify them as being alive? They're causing us all lots of problems at the moment.
That's if I go to a boring barbecue, I like to trot that one out.
Well, I hope you get some interesting responses to that.
Or slap. Michelle, waterfowl, because they move all over the world with their migrations, and then you've got nomadic species, really opportunistic species, especially in environments like Australia where there'll be no water for years and years and years. And then all of a sudden there's lots of water and waterfowl just appear from everywhere. And allied tribes like the waders or shorebirds, whatnot. Are there concerns that we could have a really major breakout of a virus? We can talk about influenza specifically, but that the wild populations are significantly at risk from farming practices?
So I guess let's start just by talking a little bit about avian influenza viruses in Australia and farming practices in Australia. So Australia, we don't have an enormous poultry industry like you see in other parts of the world. It's actually quite small. I think that North America, for example, and Asia produces far more poultry than Australia does.
Well, I was really thinking about Southeast Asia, where when I've travelled there, everybody is farming ducks. Everybody's got some chickens, and the popularity of cage birds in some countries is still huge, and a lot of those cage birds are wild caught. So that's really where my question was going, where you have people with wild caught cage birds and their poultry production really close by with the fringes of forests or living on the edges of rivers. Are we concerned that we could be the cause of a massive pandemic for birds?
Look, there's always a cause of concern for that. Avian influenza is a great example of that. We've seen these big outbreaks in wild populations due to these viruses that have escaped poultry production and gotten back into wild birds. It's not something we're concerned about. This is something that's happening. We're seeing these mortality events related to highly pathogenic avian influenza virus. We've seen it in Europe for a few years in a row. It's been seen in Asia. So it's happening. And there's always a risk that viruses that are in poultry production will spill over into wild birds and cause mortality events, because there's a number of viruses that are in poultry. For example, another great one is Newcastle disease virus. It's a really old virus. We've known about it for a really long time, and it's notifiable in Australia. If we find it in wild birds, we would have to report it because there's concern that something like that gets into wild birds.
Can I just stop you there? Newcastle disease, I think all of us have heard of it, but what is it? Like, how is it different from the flu? Is it a respiratory disease as well? And does it have like an equivalent in humans to make it easy for us to understand the effects of Newcastle disease?
Okay, so in birds, something like avian influenza, in wild birds, it's actually not respiratory at all. It's the virus replicates in the gastrointestinal tract, which is why we take fluid as well. But in poultry, you do see it as respiratory and gastro. And so influenza virus is caused by a virus called avian influenza virus. It's part of the orthomyxoviridae. So Newcastle disease is caused by Newcastle disease virus, which is now called avian ortholeucovirus 1, and it's a paramyxovirus. So both these viruses use RNA in their genomes, but they're different families. They're quite distantly related. So in wild birds, like avian influenza, there's a version of these paramyxoviruses don't cause disease. Again, we detect them really well with colloquial swabs, but in poultry, they can be respiratory and gastrointestinal. So there is a human paramyxovirus. It causes the common cold. It's called RSV. It's pretty common in kids, but it's not.
Is that commonly called rhinovirus?
No, that's a group of picornaviruses. Retinosuccinctal.
Okay.
example. Yes. And there's lots of different paramix. In fact, we've found other kinds of paramyxoviruses in penguins too. Going back to the start of this conversation, we found three different paramyxoviruses in penguins a few years ago.
And look, Michelle, I'm really old. I can remember that we had a virus called myxomatosis, which went through the rabbit population in Australia.
Yeah.
Now, of course, the non-scientist in me hears mix. When you're speaking, you're saying mix. Are we talking about a similar virus?
So no, don't worry. Look, these things don't matter. So there's a lot of different viruses out there. They are classified the same way you classify anything else, with sort of genera and families going up, and they're very diverse. And one host, so either people or birds, would have lots and lots of different kinds of viruses. They may infect the same part of the host, but be from really different families. So if we think about the common cold, that includes 4 different coronaviruses, lots and lots of rhinoviruses, RSV, could be influenza. There's a whole bucket load of things that we call the common cold because we feel crap by it. And it's pretty common. So ducks, wild birds, poultry, they also have a whole bucket load of things that affect.
So we don't have to be really imminently worried about a global pandemic for birds. I don't know if you are aware, I'm sure you were, those stories of the birds recently, what are we talking, maybe 5 or 6 months ago, just dropping out of the sky in North America. Yeah, do you remember that? There were hundreds at a time. People were just finding these dead birds all over North America.
Yeah, so just before we go on to that question, I just want to go back to your question about just bird pandemics. Every country has a surveillance system for viruses of concern. Okay, so in Australia, we actually screen thousands of wild birds for influenza every year. There has been screening programs for other viruses as well, like Newcastle disease. So it's not to say that we're not concerned about virus outbreaks in birds. We are. And so most countries do work to do surveillance to understand what's there. So just to make that be clear.
Yeah. I think I was saying we're not panicking.
No, we're not panicking, but we are paying attention. We are doing that. And I work with a lot of sort of jurisdictions in Australia and great collaborations with different partners in government. And there are surveillance systems, and we do notify them whenever we find interesting viruses that they should be aware of. There are national programs that are coordinated and things like that. So we are paying attention. We're not panicking, but we are paying attention.
Now, who's managing those programs? Is it departments of agriculture? Is it things like AQUIS, or are the universities managing those programs?
So the program that I work with is mostly the National Avian Influenza Wild Bird Steering Committee. And it's myself and Marcel Claassen from Deakin University. There's a number of sort of state and jurisdictional government labs that are there. So, for example, DPI New South Wales, AgriBio Victoria, more acronyms than I can think of. And we work closely with Wildlife Health Australia. Okay, so there's somebody from Wildlife Health Australia on there. We have seats. I know that BirdLife Australia is always welcome to attend calls. The egg industry are welcome to attend. So a broad group of people that we share data.
So it's a quango. So it's a quango overseen by, is it reporting to the Federal Department of Environment or Department of Primary Industries, or?
So we have a Department of Agriculture. So because the Department of Agriculture, Commonwealth Department of Agriculture provides some money for the program to do the surveillance, and then all the different state labs and jurisdictions as well.
Yeah. And well, because we're not really hearing of outbreaks regularly, must be doing a good job there.
I'd like to say that's the case, yes. I think everyone is doing their best and working well together. But Australia is also quite an interesting place for viruses. And this goes back to something I think you'd asked before that I'd never quite got around to answering. And that has to do with bird migration. So if you think about North and South America, there are a lot of birds that migrate through those regions. Europe, Asia, and Africa, there's a lot of birds that migrate through those regions. But other than waders, so shorebirds, there's not a whole lot of ducks that migrate. Actually, there are no ducks that migrate out of Australia into Asia past sort of Papua New Guinea. You may have heard of the Wallace's line. It's a pretty important line. Yeah. And so one of the hypotheses is that because there's not a lot of ducks migrating across there, we're pretty isolated here. And so there's not regular introduction of viruses like highly pathogenic influenza virus from Asia into Australia. We just don't really see those introductions as well. This is not to say it can never happen. Again, we're paying attention. We're doing surveillance to ensure that if it does, we catch it. But it means that I think we're, of all the places in the world, hopefully we're at a lower risk of some viruses due to these interesting dynamics of how bird migration works here.
But of course, there's the other side of that coin, isn't there? That our birds are not co-evolved with a virus that may be introduced accidentally or by some straggler or migrant from some far-flung part of the world.
Influenza has been in Australia probably for as long as birds have been here. These low pathogenic avian influenzas are found in birds here, and a number of different viruses, wild bird viruses, are found here as well. So it's not to say that we don't have viruses, the same as Antarctica. It's not that Antarctica doesn't have viruses. It's just that we tend not to see a huge introduction of sort of some of these poultry adapted viruses that are common in other parts of the world being introduced here.
So it's about 9 years ago that you were down in Antarctica, on the Antarctic Peninsula. Whose base were you at on the Antarctic Peninsula?
So I went with colleagues from Chile. So Daniel Gonzalez Acuña, who unfortunately passed away last year, was the team leader. And I was working in Sweden at the time, so we had a collaboration with his group. And so we went with INACH, which is the Chilean program. And so we stayed in Chilean and one Argentinian base while we were there.
And what's the best thing about working in Antarctica?
I thought it was really cool. Those things you watch on documentaries that in real life, they're never quite as good as they are in the documentaries. Well, this was better. One of the places we stayed at doesn't have a big human presence. There's just like a tiny little old base. And you walk around and there's spur seals there and leopard seals hanging out. And you can watch all these seabirds right from your breakfast table, and all the penguins are making a ruckus just around the corner. It was really cool. It was such a great privilege to be able to be there and to be able to see that. I think that's really special.
Yeah, I meant to ask Elodie this when I talked to her about her time at Kerguelen, but you'll do. How stinky are penguin colonies?
I think that penguin colonies are pretty stinky, but I had worked with seabirds for a number of years before, so I didn't necessarily find it much stinkier any other place that I've been. But I will state that my coveralls that I used in Antarctica stayed in Antarctica. I did not bring them home because they were, well, what do you expect? You get millions of individuals coming together, highly congregated place. They're sitting on nests and they're defecating around. That's just it being stinky is part of the game.
And see, one of the things that has occupied my mind while we've been bored out of our brains in lockdown is what's the stinkiest bird in the Antarctic? Is a penguin stinkier than albatross? Or
well, I think it's more a question of critical mass, right? Because there's a whole lot of penguins smushed together in a very small area. I didn't work where there were albatross, unfortunately. But, it's the same with working in seal colonies. They're pretty stinky because there's just a lot of animals in a really small space.
So what were the penguins that were on the Antarctic Peninsula? Who were your best penguin buddies?
So we mostly our project aim for that year was to work with chinstrap penguins, but we were, of course, surrounded by lots of Adelie and Gen 2 penguins down there as well, which was really.
And which are the coolest penguins?
I think Adelie penguins are hilarious. They have quite a lot of attitude. They're very funny. Probably also like penguins other than chinstraps, because we work with chinstrap penguins and I sat on the arse end of a lot of chinstrap penguins and got crapped on by a lot of them. So we're going to pick Adelies because they didn't crap on me so much.
How similar are those three species in their behaviours?
So I'm not a penguin expert. I know about viruses of penguins, but I'm not a penguin expert. So let's just start with
Just
that.
personal observations of casting your eye out over the colonies.
They are a bit different. I think I found Adelies were doing slightly different things from the other two penguins, and their colonies were often in different places. So a lot of the islands we visited had both chin straps and gentoos, but Adelies were doing different things. And from a virus perspective, based on very limited data, they seem to be doing different things as well. We see lots of viruses in Adelies and much more connectivity in colonies by Adelies as well. Yeah. But again, not a penguin expert.
I really just wondered whether the gentoos and chin straps pinch each other's rocks from their nests. You can never get sick of watching that.
I don't think I saw a lot of thieving across species. There was a lot of thieving within species for sure, but not so much across species. But they were always really funny to watch. I really enjoyed watching them do their thing.
Yeah. And what's the voyage like? Did you depart from Chile? Like you were in, because this is really interesting. You're in Sweden. You've applied for this position, no doubt. So how long did you have to prepare, and where did you depart from?
So I actually went instead of somebody whose PhD was about penguin or viruses, bacteria. I think he was working on bacteria in polar regions, and they were collecting virus data as well. And I think he had finished his PhD and was seconded back to his job. So there was an opening on this team. And so I was just in the right place at the right time, actually. Unlike Elodie, who had to compete very hard for her position, I was just in the right place at the right time. And so I think I have remembered having a conversation in the autumn and then going I think we departed in January, early in January. So it's not like it was quite a long process. Again, really different from Elodie and probably different from a lot of other people's experience. So yeah, it wasn't such a big thing, which is a terrible thing to say.
That's amazing. I mean, that it's almost like a spur of the moment thing that somebody can't go, and then casting around and go, "Oh, Michelle, are you interested?"
Yeah, absolutely. I don't think I'll ever get an opportunity like that again in my life. I think I was really lucky, and I went down as the virologist of the team, so that was really good. A good place to be.
Did you depart from Chile?
Oh yeah, sorry. So I flew to Punta Arenas, which is in the southern part of Chile. And then we actually flew in a plane to King George Island. So the Chileans have a runway on King George Island. So we flew in a big plane. And then we were escorted between bases using Chilean naval vessels. So the Chilean military. And so I didn't have one of these great big passages across the Drake like most people do. I actually flew in and out of Antarctica. Yeah.
And how long is that flight? You went from King George Island to the peninsula on a boat. So on a ship. How long was that last stage, King George Island?
So I need to think. It's a really long time ago. So we went from, I think it was probably less than a day. I don't remember being on the boat for very long. So we got picked up from King George and went to Livingston Island first for 2 weeks. And that was not a long boat journey at all. And then we went from Livingston Island to O'Higgins Base, which is on the peninsula. And I think that was probably a day at most. And it's all through the island. So the sea is quite calm and flat. I remember having incredibly beautiful weather. Like it was like glass when we sailed through there. And then we were at O'Higgins Base for a little while and then went back to King George. And again, that was probably only a day.
So how active are the Chileans in their research in the Antarctic zone?
Daniel Gonzalez Acuna, who I worked with and went with, has been working in Antarctica for many years. So he had been going for a few years prior to when I went. And he continued to go most years after. And like I said before, unfortunately, he died last year. But I have seen from the literature that there is another group in Chile who are starting to do more work in Antarctica. And they seem to have been collecting samples for the last few years. So I know that INACH has a big competitive process to be in Antarctica. So I think that the Chilean researchers are quite active down there, but I don't have a good overview of the whole project.
I wondered whether it was...
Most countries have scientists. There's a lot of research to do, and because so many countries claim sort of pieces of the pie, they'd like to be working there. And so we see all kinds of research from all kinds of countries that have bases down there.
And the Chilean base is staffed by its multinational population, isn't it? It's not mostly Chileans. They just pick the best people to work on whatever they're working on, yeah?
So we mostly stayed in military bases, and there is a very big military presence in Chilean military presence in the Antarctic Peninsula and islands. And so when we were there, we were usually the only group of scientists at the base. Maybe there was one other group of two, but there would have been a very large military contingent when we were there, and they were all Chilean. Yes. I didn't have sort of the experience that a lot of people would have had going to primarily research stations that were really multinational. Through the Chilean program, we went on military vessels and stayed in military bases.
Okay, I'm not sure that's generally understood. Certainly not the Australians. I think we think that the military is hands off in most of the stations in Antarctica. I mean, of course, the military fly people in and out and ferry them around and whatnot, but I wasn't aware there were garrisons down there.
Yeah, so I think the difference is that the Antarctic Peninsula is claimed by like 6 countries or more. I don't quite know. There's a lot of countries claiming the same slice of the pie, whereas the Australian territory is largely undisputed. It's Australian. And so I think my interpretation of the situation, I may be wrong, but is that in the Antarctic Peninsula, there are lots of military garrisons by a number of countries because they're all trying to claim that slice. And so what I had heard at the time, whether it's true or not, so one country built a base, then another country built a base, and then one country sent down a pregnant woman so that their child would be the first baby born in this country. And then the next follow-up was they sent a couple down to conceive and be pregnant and have it. There's a lot of jostling to claim pieces of the pie down there. So
for those really important milestones.
It was pretty odd, but I had a great time and we were really well supported and taken care of. So it was a really great experience.
Oh, I envy you. I think it would be marvellous. So how did you end up at the University of Sydney? It's a long swim.
Yeah, so I think one of the really fun things about being a scientist and working in science is that my qualifications and experience are relevant all over the world, meaning that I could get a job wherever I wanted. And the result of that is I've worked in quite a lot of countries. So I'm Canadian. I did my bachelor and master's degree in the eastern part of Canada. I've done field work in Alaska in the Aleutians, which was really fun. And then I did my PhD in Sweden and did my sort of first postdoctoral position in Sweden. And then my position was ending and a job came up in Melbourne on avian influenza in wild birds. And so I interviewed for the position and was accepted. So then I came to Melbourne in the very end of 2016 and worked at the WHO Collaborating Centre for Influenza in Melbourne, working on avian influenza viruses. And I did that until this time last year, where I moved to the University of Sydney with an ARC fellowship. So a bit of a long, convoluted journey.
Okay. And I try and explain to the general public, like the bird nerds who aren't in academia, what's a fellowship? What does it entail for you? And is it time limited? Give us an idea of what life as a travelling academic is like.
So the academic job market, it's a bit of a racket, actually. There are very few permanent positions and very few positions paid. Your salary is paid by the university. A lot of positions are contract positions and your salary comes from like money from a research grant. So for example, my current position, I applied to the Australian Research Council to undertake a project. So I asked for 3 years of salary money and running costs in order to undertake this project. And it's very competitive. I think about 10% or less of these early career fellowships are funded by the ARC. So I was really lucky to put in a competitive application. And so my current position is funded through grant money on this fellowship. My previous position was also funded from grant money. So 2 years from a sort of project money, and then the next 2 years were combined money from projects we put in and contract work we put in and some other bits and bobs.
When you describe it like that, sorry for interrupting, but you had 2 years funded from grant money, and then you have to scrabble around for bibs and bobs for the next 2 years. This is the point I want to get across to people, because we have this terminology, all these people in their ivory towers, in those universities and whatnot. But you do amazing science, but there's no tenure, there's no job security. Let's be honest, the wages are pretty crap. And every couple of years, you've got to start the whole process again. And it takes you about 6 months to write a decent proposal, and then you've got to wait. And in that time, you have no security whatsoever. So
no, and so one of the reasons many people move all over the world is because their job has run out and they found something in another country that is similar to what they're doing or what they're interested in, or they put grant money into or a proposal in to work with someone somewhere. So yeah, there was a good decade where people move around a lot and are on very tenuous and short-term contracts, but doing a really amazing science.
Yeah. And contributing a lot, because are you teaching as well?
I'm not currently teaching as part of my position. I have a research-only position.
Yeah. Again, I don't think that the wider community understand this, that you're in precarious employment, the tenure is not sure. But then because universities don't employ anyone full-time, it's presented that it's a great opportunity for you to supplement the shit money that they give you for it. But it's just a way of not employing more people. The information transfer is fantastic, and the fact that you have worked all over the place and have experience to impart and everything is great. But the downside of it is that you don't get the opportunity to have that sort of permanence in your life that somebody who is a sheet metal worker might have. Everyone's job is precarious. We understand that. But your whole industry operates on precarious work. So when do you get to decide that you want to stay somewhere? Like,
I guess when you decide to stay. For lots of people, they will do these precarious contracts for a few years and then decide they've had enough, and they'll leave academic science to pursue something different, whether that's in government, private industry, or a whole different career line. I think I was just speaking to my partner about this the other day. I think it's really sad that a lot of people who are in my cohort, really stellar PhD students who did amazing research, there's very few of us left in academic science. Most have gone on to do other things, which is good for them. They get salary and stability, and that is what people need in their lives.
Absolutely. But look at the wasted. We talk about corporate knowledge when companies downsize and sack everyone, and all that corporate knowledge walks out the door. We've designed that into our academic system. It's just seems a little bit bonkers that we invest so much in bringing people up to be highly trained, great pools of knowledge, and then it goes out to the private sector and someone owns it.
Yeah, pretty much. And especially with the pandemic, universities were not eligible for job keeper, and so they paid huge numbers of staff in last year by especially teaching staff, people who are paid like me by grants. We can keep our positions because our salaries are paid by grants, but people that were paid by universities, especially teaching staff, have had a really tough go of it last year. And so I think it's been very hard on the academic sector in Australia for sure.
And of course, there'll probably be a shakeout as universities have to realign. And of course, the government has changed the priorities for funding certain types of courses. So who knows where that's going to leave. I think pure science is going to be okay, but I'm not too sure about applied science. But fingers.
Yeah. On this point, there's just one point I would care to make. It's not so much about birds. It's more about the fact that in Australia, I think it's really important to remember that most people working on the pandemic response are in the university sector. There are a huge number of people who worked on the University of Queensland vaccine when that was in progress, people who are looking for antibodies. There's doing all of these people are on tenuous contracts in the university sector, and there's no investment in keeping them. So that to me is cool. As the last thing to say on that topic.
Nearly the last thing. So, dear listener, if you're wondering why I'm so interested in this, I do another podcast called Migrating to Australia, and and in that, again, I'm the dumb sidekick to an immigration lawyer, and this issue frustrates me over and over again when I see how difficult it is for people in your situation, academics who have been doing, especially research academics, and you find it really hard to put down roots in this country. You basically have to marry an Australian if you want to stay. So it's become so hard. That's pretty much the option, which I just think it's short-sighted. But I don't think Australia is on its own like this. I think that there's this eternal churn, which is an opportunity for a lot of people, and that life suits them. But there comes a time where that life isn't quite so appealing for a lot of people. And I just think it's a shame that a lot of that knowledge is lost because the career change is complete and total. It's very hard to stay on the fringe of research and practical work, applying your skills. Yeah, I think there's a lot of people who are now designing kitchens and things like that using skills that they could be applying to saving endangered species, which is why I bang on about it all the time.
Yeah.
So what's your sort of next move, do you think?
I don't know.
Yeah. I haven't done that career planning yet, Michelle.
I have 2 years left my fellowship, and I'll let you know when I know.
Well, part of the question earlier was what I like to ask people where they see as their 5-year horizon. What would you like to be doing next if you got all your ducks in a row?
If I got all my ducks in a row, so to speak, I think it would be great to get a junior position, so to have my own lab, have my own students. Yeah, that's hopefully where I'm going. Let's see how we do.
And is that the kind of position that comes up regularly, or do you have to wait for someone to retire? Or
so again, I think the good thing about being able to go anywhere in the world is I'm not fixed to waiting for someone retire in a city or at one institution. Positions do come up. Whether you like the institution or not is a consideration. Whether the institution likes your portfolio is a consideration. And whether you're competitive or not for that kind of position is a consideration. So they do come up from time to time. So you just got to keep your eyes out and try to land a good place.
Okay, well, the deal that there's the bird emergency questions. So let's do them. What's your field guide of choice?
So I have a Simpson a day. And the reason for that is before I moved to Australia, I felt the need to buy a bird book, and the only Australian bird book in the shop in Sweden was Simpson and Day. So that's what I got. My partner has, I think it's Morecambe. And so usually when we go out birding, we have both of them together, and I have the Morecambe app on my phone.
And when you go back to visit your family, I'm assuming they're still in Canada. Mm-hmm. What's your favorite book there, eh?
I think I have the National Geographic Birds of North America, and I have the Collins Guide to Europe when I'm back.
Oh, very good. And have you moved across to using any of the apps?
Yeah, so I downloaded the app last year, actually. I'd never used an app before, and we were in the Victoria and Mallee in December, and I was really keen to go look for Mallee emu wrens, it sounded like. I was hanging out with, what's his name? I think, is his name Michael? I can't remember now. Explore the Mallee. They've got a little place, and so he put me onto come out. Yeah,
might have been name checking another episode that's coming up because we've got a
No.
Melly
What's
Emu
it
Wren
mean?
episode about to.
Yeah. So anyway, I downloaded the app because I needed to figure out what these things sounded like so that I could actually find them. And I find now that my local bush birding, I find the app really useful because often I'm 90% sure and just need to quickly check to make sure I'm 100%. But when I go to a new place, I much prefer a book over an app. Yeah.
So, is the app now your favorite piece of equipment when you're out in the field? Or is it the trusty binoculars, or is it your wide brim hat?
That's probably my binoculars for sure.
And are you using a little pair, a big pair?
I have a Swarovski 10 by 42 that I've had for like 10 years. Yeah.
You hear that, Mr. Swarovski? Looking for a sponsor.
They're great. They are great binoculars. Before that, prior to that, I had a pair of Bushnells that I had bought when I was like 9 years old. They were these tiny little.
Yeah, and were they the rubber coated ones?
Yeah. And so when I bought the Swarovskis 20 odd years later, it was like night and day. These binoculars were amazing. The amount of light you get in and how far you can see, it was really amazing. And they've been on a few continents and have had quite a beating and are still in good shapes.
Oh, that's good. And what about the little pair? Are they still at the bottom of the backpack, just in case?
I don't know what happened to them, but they did not move with me to Australia.
Handed them off to a cousin or something, a little cousin.
I don't even know.
Now, you've worked with so many interesting birds, so I'm interested to know what your favourite bird is.
I think that seagulls are entirely overrated, or underrated, actually, what I mean to say. I like gulls, and I like them because they're very funny and very smart and quite fun to look at. I spent a few years trying to catch gulls in North America, and I never caught the same individual twice. They'd come to the edge of the trap and look, but you'd never catch them again. They're very smart. They're very feisty, and they have great personalities. So I really like gulls.
So how does the silver gull compare to? Oh yeah, was that a Pacific gull or a herring gull? So how does the Pacific gull line up against the herring gull?
Pacific gulls are great. I really like to see them. They're really beautiful. We've universally caught silver gulls a few times in some of our nets, and they're also really feisty, and I really like them.
Yeah, so I'm sure silver gulls think they're pigeons.
They think they're pigeons, but I think they're bigger and meaner than what they are.
They're actually stunning when they're in good condition.
Yeah.
And again, you've been in so many locations, great locations. Where's your favourite location that you have been or do go birding?
Birding? Okay, so birding, my favourite place to go birding is the northern bits of Botswana. It's really lovely. I don't get there very much anymore, but I've been to the parks a few times. So Chobe, Savuti, Muremi, all those parks. Because the bird diversity there it's incredible. You can easily see, I don't know how many birds we saw, 200 birds over 10 days or something. It was really amazing. So international birding, that's probably probably the top place to go. I love babblers. They're so funny. The African Aramark babblers, you hear them before you see them, and they just come in and they're all fighting with each other and making a ruckus. And they're great. And babblers are very funny too, though, but they're a bit harder to see.
And have you got a favourite spot in Australia? You've spent a lot of time in Melbourne and now a lot of time in Sydney. Have you got around to many other spots in Australia?
So we went birding in Kakadu in 2019, which was great. What a great place. How amazing. And a Western Treatment Plant is in Melbourne. Western Treatment Plant is a great place to go birding. Yeah, but I think Australia is great to go birding everywhere. We go to all kinds of places to see things. Yeah.
Yeah, you haven't got a favourite place you go back to time and time again?
No, Treatment Plant I've probably been to the most times. There are quite a lot to go catching with the BWSG, so it's a pretty regular place to go. But I don't think that there's a spot where I go time and time again. Yeah.
Now, I'll just translate that for the international listeners, that Western Treatment Plant is a big sewerage farm that has this great reserve, great area of reserve, which is really what do we describe it as? Some people say it's a bit of a heath, it's a marsh, it's a grassland. It's a bit of a mixture. Lots of those sort of succulent plants in some areas of it. So that's one of the favourite spots for urban birding in Melbourne. And you can see some pretty interesting things there. Waterfowl, shorebirds, lots and lots of passerines. So, yeah, terrific place. And you mentioned the acronym for the Victorian Wader Study Group. So, yeah. And you'd be amazed. I get messages from people going, I didn't know what you're talking about. Are you a bander?
Yeah.
Yep. So, a registered
but I don't get to do a lot of banding because usually when we're out in the field, I'm taking samples for research. So it's actually been a number of years since I've done a lot of banding, dedicated banding. Yeah.
It's pretty hard to be a bander. You got to be good.
Yeah, well, I learned I did a lot of banding when I was in Canada because I did a lot of field work on my own and with one other person. So it wasn't as nice as it is in Victoria, where we go and work with teams of people, where there are many other banders there. So yeah,
I've got to throw in an extra question. What's the best bird in Canada?
I did a lot of seabird work when I was in Canada, and so I'm probably going to have to defer to seabird. I got to work with puffins, Atlantic puffins, when I was there, which was they're so great, and we call them guillemots in Canada. Murres in Canada, they're called guillemots in Europe. Yeah. They were so lovely to work with, and razorbills are great.
And I'm always in awe when I'm watching bird Twitter. The things like buntings and warblers in Canada, there seems to be, and North America generally, seems to be an amazing diversity. Do you go out looking for those bush birds?
So when I lived in Canada, I spent most of my time on the northeast corner, which is the very end of the Atlantic flyway, and we did not get. So I lived in a place that had a lot of seabirds and icebergs and not a lot of warblers. So when my parents are Vancouver's, when I would go visit my parents, we sometimes look at warblers, but I wasn't often there during migration.
Northeast, that's sort of Halifax, St. John.
St. John's, Newfoundland is where I live. Yeah. So there's a big old island. You got Greenland, and below Greenland you have another island called Newfoundland. It's part of Canada. A lot of icebergs.
And big dogs.
Fantastic.
So where's somewhere you really want to go birding?
So our next Australia destination, if we can get there, given pandemics and lockdowns and things like that, is Cape York. I'd really like to go and see eclectus parrots and cockatoos. So hopefully soon.
Hopefully. And what's your bucket list bird? Have you just mentioned the two of them, or is there something that's at the top of the list?
I don't really bird like that so much. I think I'd usually rather just go to places and see whatever is there. It's very rare that I like to go hunting for a bird. We'd obviously like to see things like fallen cockatoos and eclectus parrots if we go to Cape York, but if we don't see them, that's okay because there's lots of other things to see. So I'm not a big twitcher, just a casual birder. Yeah.
You've answered that question, and you can't tell me your number, your life list number.
No, I have no idea. But the number is probably pretty high, given I've done a lot of birding in Southern Africa, and I did a bit of birding in South America and North America and Europe, a little bit in Asia and Australia. So I don't know.
It's going to be a lot more than mine.
Not necessarily. Like I said, I'm not a big twitcher. I'm a very casual and relaxed birder. So I've probably been a lot of places and missed a lot of things, and that's okay.
Oh, that's definitely okay. It's good. So you're down the fun end of the spectrum, and that's where we like to see you. And I guess the last thing I'm really interested in is, do you think you'll be birding for life?
So from what I hear is I started birding when I was a baby. I had a short hiatus for a few years where I didn't bird, but I think it's something I will always do to some degree, whether it's more seriously or more casually. It's a great hobby. It's nice to be outside. Sometimes I say I'm a bit of a naturalist. I like to know the names of things that are around me. Obviously, Australian plants are really hard. There's so many eucalyptus, but and so I think it's something I'll always do to some degree, no matter where I am. And even if that means looking at the miners outside my balcony, I guess that's still bird watching in some way.
Correct. I saw a lot of miners in the far-flung reaches of the Philippines, different miners, and sparrows where they actually weren't an introduced invasive species, where they were hanging out where they were meant to be. And I have a whole new respect for those birds, which as a kid I hated with a passion. Here and woke me up that birds are just doing bird things. It's not their fault they're here.
The reason they're doing well is they're adaptable, and they deserve some respect for being able to survive in a totally new environment and doing well in it.
Absolutely. Miners are very funny birds.
Oh, they're hilarious, actually. I like to watch them. They're pretty cheeky.
Bloody invasive buggers when it comes to nest hollowers.
Yeah.
Well, Michelle, thanks a bunch. Now, if someone wants to check out some of your writings, your adventures, where should they head?
Well, so I am on Twitter. I tweet things about birds and viruses. So you can find me at DuckSwabber, which is a pretty memorable Twitter handle. Yeah, otherwise my academic work is on a number of academic sort of websites, ResearchGate, Google Scholar, and things like that.
Yeah, no, I think your Twitter handle is up there with Steph Burrell, who has got Petrol Station.
Ooh, that's good.
That's a ripper. I think you're up there. We got to the end of yet another one. Thanks to your listener for hanging on. Thanks, Michelle. Thanks so much for being a part of it. And, well, happy lockdown, happy pandemic. And may there be many more viruses for you to swab out of ducks' bums. So,
Thanks for having me.
Bye.